Hi everyone, I'm really glad to be here today and to have the occasion to present to you my latest work, which title is Share Reorganization Across Sensory-Deprived Individuals, Review Higher Plasticity of Visual Cortex. Over the last decades, the quest on the characterization of the sensory-deprived brain has offered scientists a unique opportunity to understand to what extent a sensory experience, or the lack of it, shaped the development of the brain functional organization. In fact, evidence exists that the human brain develops its large-scale architecture even in individuals who lack sensory input since birth, and that brain organization is primarily driven by property-specific and modality-invariant computation, a phenomenon that we call supramodality. Recently, we expand this notion of supramodality even to multisensory brain regions. In fact, in a recent work, we measured brain synchronization during the presentation of different versions of the same narrative in distinct groups of sensory-deprived participants and typically developed individuals. We enrolled congenitally blind and congenitally deaf subjects, and we presented them with the auditory and the visual version of 101 Dalmatian movie, and then we measured the intersubject correlation. We reasoned that any evidence of synchronization across condition and experimental groups will be indicative that the specific regions yield a shared representation across auditory and visual features despite so different postnatal sensory experiences. Intersubject correlation analysis revealed that the superior temporal cortex was synchronized across condition and also across groups, as the image on the top part of the figure displays, even in individuals who lack any audiovisual experience, and so irrespectively of postnatal sensory experiences. Therefore, we conclude that the human superior cortex is innately provided with a scaffolding to yield basic multisensory representation and to perform also coherence across sensory events. And this is what we call the proto-organization, using a term originally used by Arcaro and Livingstone in their work. Nonetheless, because of the lack of a sensory modality since birth, individuals undergo also a plastic reorganization in the deprived cortices that retain therefore the ability to process the non-preferred modalities. However, it is unknown whether different sensory systems entail different degree of plastic reorganization in the human brain. In fact, the degree of experience-dependent plasticity in the brain is non-homogeneous, and there are remarkable shifts in plastic potential that can be observed mainly due to time constraints, for example to sensitive periods, and also to the hierarchical level of processing of the function that we are considering. Indeed, within the same sensory system, different functions can have different degrees of adaptability to the environment. Plasticity for primitive function tends to close earlier than plasticity for more complex one. And the aim of the present study was to test the prediction that visual and auditory regions do actually display a distinct plastic potential. And to assess this idea, we measured to what extent alteration in the sensory experience affect the functional architecture of these two systems. Therefore, we used the same dataset that we acquired for the previous study that I showed you, which is actually an fMRI experiment with three basic experimental conditions. One that is the audiovisual with the full presentation of the movie, so multisensory presentation of the movie, in which we recruited 10 typically developed participants. This condition is important to us and serves as a benchmark also for brain parcellation, as I will show you in a moment. Then we have two unimodal conditions, an auditory-only and a visual-only condition that were presented to four groups of subjects, so typically developed participants and congenitally blind and congenitally deaf subjects. For what concerns the analysis, we perform a within-group intersubject correlation analysis for each group separately, and then to address our question, we measure whether difference in intersubject correlation values do exist. These are the results of the within-group intersubject correlation for each group. As you can see, the whole brain analysis was computed for each experimental group separately. As reported in the figure, movie listening elicited a significant intersubject correlation value within a cluster of regions that are actually devoted to the encoding of auditory information. That includes primary and early auditory cortex, as well as high-order cortices belonging to temporal and parietal lobes in both congenitally deprived and typically developed participants. The bottom two rows instead represent the intersubject correlation maps during movie watching for both the typically developed and congenitally deprived subjects. And we can see that here there is a much broader recruitment of the cortex with the synchronization that spreads across occipitotemporal region and frontoparietal areas. We can also notice that there are some differences as well among groups. The first that catches the eyes is the one between blind and typically developed people that listen to the movie. Indeed, blind subjects recruits the calcarine fissure bilaterally and some region of the inferior temporal cortex to process auditory stimulation. And this is a phenomenon and a result that has been already reported by other studies and other groups. Here I will just deviate a little bit to introduce the methodological aspect that it will be important to understand the result of the intersubject correlation differences across groups. Indeed, we took advantage of functional gradients to study the functional organization of the brain. In fact, gradients have been shown to capture the large-scale principle of cortical organization as well as also modality-specific granularity, providing therefore a more fine-grained way to interrogate and understand the principle of functional cortical organization. Therefore, we concatenated our functional time series during movie watching and we averaged them basing on their assignment to a cortical parcellation. We use here the ACP Atlas from Glasser and colleagues, and then we computed the similarity metrics that capture the inter-area similarity in functional connectivity during audiovisual stimulation. These affinity matrices were then decomposed using a technique that is called diffusion embedding, that is a nonlinear dimensionality reduction algorithm that yields a set of manifolds or gradients for the movie. And this is important for us because we will map our results on this 2D representation. So I will briefly guide you to reconstructing back the projection of the brain cortical areas onto our 2D representation of the brain. So in red you can see late and early visual regions. In orange and yellow, posterior parietal and superior parietal cortex. Again, sensory motor regions are represented in yellow here. Then we have inferior frontal and superior frontal regions that are mapped in green, as well as early auditory areas that includes A1 and belt and parabelt regions. In blue, we mapped inferior temporal and superior temporal regions, and in dark blue, orbitofrontal areas. Okay. So these are the results for the difference in intersubject correlation between blind and typical developed subject during movie listening. We used the 2D representation that I just showed to you, and with triangles we indicate higher intersubject correlation in blind participants, while squares were used to indicate lower intersubject correlation in blind participants. Results show that blind individuals synchronize occipital regions significantly more than typically developed participants, as shown by the triangle in red in the upper portion of the image. On the other hand, synchronization in early auditory, superior temporal and frontal regions is lower in blind than typically developed subjects, as you can see from the greenish squares on the bottom part of the image. These are the results for the difference in intersubject correlation between deaf and typically developed during movie watching. Deaf individuals synchronize the occipital and posteroparietal regions more than typically developed participants for both hemispheres, as you can see from the triangles on the upper part of the image. At the same time, you can see that deaf individuals were less synchronized relative to the typically developed in the superior temporal region. No differences in synchronization emerge in primary and early auditory cortices. To summarize our results, we observe both patterns of greater and reduced synchronization profiles in congenitally blind and deaf participants compared to typically developed individuals. Importantly, occipital regions show greater synchronization in both congenital visual and auditory deprivation relative to typical development, and this suggests that visual areas are reliably involved in stimulus processing regardless of the deprived sensory modalities. Overall, in both models of sensory deprivation, we found a decreased engagement of the temporal areas. These findings support the hypothesis of an intrinsic organizing principle in the human brain in which the occipital cortex undergoes a greater degree of plastic reorganization than temporal regions. From an ontogenetic perspective, this finding is consistent with research that show that sensitive period for the visual system lag the auditory one, and in general with the evidence that the visual system express a more protracted functional refinement and maturation. I want to thank all my colleagues that work in this project and all of you for your attention. Please feel free to contact me if you have any question or any curiosities. Thank you again.